Okay, ladies and gentlemen, good day all. Today we are going to look at the concept of metabolic regulation at all cell level. When we said metabolisms generally is a sum total of all biochemical reaction in the body, and these reactions is what actually make life processes in the living system. So, what we need to regulate the metabolic pathways. So therefore, in this point in this video, we are going to look at how metabolism is being controlled, as there are thousands of many chemical reactions in the living system. So in this video, we are going to see how these metabolic pathways are controlled. Because if there is no control in metabolic pathways, there are a lot of problems, and we are going to see the problems also at the same time. So, what is metabolic regulation? Metabolic regulation is a term that used to describe the process by which metabolic pathways, and these metabolic pathways can be anabolic or catabolic. So, remember anabolic pathway are the pathways that are associated with the synthesis of biomolecules. So it means that in anabolisms, we are talking about how smaller molecules are utilized to generate bigger molecules. And that is, of course, anabolism. So in the case of anabolisms, you are using smaller molecules to generate a larger molecules, and that is anabolisms. And usually in the anabolic process, energy is usually utilized. So you need to add energy. Energy is needed to activate the smaller molecules to generate the bigger one. So also at the same time, when we said catabolism, it means it's a degradative pathways where a larger molecules will break down to produce smaller one. It will break down to produce smaller one. And of course, generally during catabolic pathways or during this degradative pathway, energy is usually generated. So at the end of every catabolic process, although not all, but in most of the catabolic pathways, energy are generated. We usually generate ATP during catabolic pathways. So you said metabolic regulation actually is a process whereby both anabolic and catabolic pathways are controlled. So that is catabolism. And why do we need to control metabolism? We need to control metabolism so that at least the products will only be produced when it is needed. There is no need to actually synthesize glucose in the body while you doesn't need it. There is no need of the body to synthesize a particular amino acid while the body doesn't need it. So that is why we have this control system where a particular metabolite will only be produced when it is needed. And at the same time, another reason why we control metabolisms, another reason apart from the production of product when it is only needed, there is issue of futile cycle. When we say futile cycles is that there is no need at the same time to be catalyzing and synthesizing the same molecule at the same time. So if you are doing that, it's like you are doing what you call futile cycle. And usually when there is a futile cycle, there will be disease, because at the end, the substrate will not be utilized in the system. So therefore, there is a likelihood that a particular cell will be injured. In fact, at the end, it can even lead to the death of the cell. So that is why metabolic process needs to be tightly regulated. It needs to be tightly regulated so that at least to prevent futile cycle. So when you said futile cycle, it means the synthesis and degradation of a particular pathway and degradation of particular pathways are taking place, of a particular biomolecules a metabolite is taking place at the same time. Like for example, glycolysis and gluconeogenesis. Glycolysis is the breaking down of glucose, while gluconeogenesis is a synthesis of glucose molecules from non-carbohydrate raw materials. So it's not possible to have gluconeogenesis and glycolysis at the same time. So one must actually take place before the other one take place. Like for example, you need to actually undergo glycolysis so that at least because you have enough glucose and you want to degrade that glucose and use it to generate ATP. While gluconeogenesis is when there is low blood glucose level or there is a low glucose in the body. And that is when some amino acids, some non-carbohydrate materials will now be utilized to start synthesizing the glucose so that at least some of the glucose will be utilized in maintaining the blood glucose level. So that is why we need to understand that metabolic regulation must be controlled because of two reasons. One, to synthesize the product when it is needed, and when the product is not needed, then it will not be synthesized. So that is one of the main points of metabolic regulation. And then second, to prevent futile cycle, so that at least a particular metabolite cannot be degraded and synthesized at the same time. And of the thousands of enzyme catalyzed reactions that are taking place in a cell, there's a probably not one that escapes from upregulation. Of course, we have thousands of biochemical reactions that are taking place in the body. And I don't think that there is any reaction that can escape regulation. Because if there is any reaction, or if there is any enzymes catalyzed reaction that escapes any form of regulation, so there will be a problem. In fact, most of the diseases that we are developing is as a result of the improper regulation of metabolic reactions. So whenever there is a metabolic reaction, then there is a likelihood that the cells will be regulated. And if there is a proper regulation in a cell, there will be no problem. So now the ability of a cell to carry out all these interlocking metabolic process simultaneously, observing every product in the amount needed and at the right time. That is metabolic regulation, is the ability of a cell to carry out all these interlocking metabolic process, all the reactions, all the ability of a cell to carry out all the biochemical reactions or biochemical process or all the metabolic processes simultaneously, obtaining only products in the amount needed and at the right time. So that is one of the main reasons of this metabolic regulation. So the products will be the amount of the products that of a particular metabolic process will be needed, and it will be produced at the right time. Like for example, let's now take examples of glucose 6-phosphate. Glucose 6-phosphate is one of the metabolites. It's one of the metabolic, or it's one of the biomolecules that usually synthesized in the body. Of course, in the body system, whenever we have glucose, so glucose plus ATP, like for example, in the glycolysis, the first enzyme is hexokinase. So in the presence of hexokinase, glucose plus ATP, it means that this enzyme, hexokinase, is using two substrates. Because the two substrate here is glucose and ATP. And these types of reaction, we call it bi-substrates reactions because we have two substrates. So the enzymes use two substrates to catalyze glucose to glucose plus ATP to glucose 6-phosphates. So hexokinase or glucokinase in the liver It's utilizing or it utilizes glucose plus ATP to generate glucose 6-phosphates. So now we have glucose 6-phosphates. And this glucose 6-phosphate has so many pathways or have so many fates that the glucose 6-phosphate can be used for. Like for example, it's used in protein glycosylation. Remember from the concept of molecular biology, gene, there is a gene, or let me say from DNA. In a DNA, we have genes, and these genes are responsible for producing a specific messenger RNA, and the specific messenger RNA will be used in synthesizing proteins. And some of these proteins, or most of the proteins, are synthesized in the ribosomes. So after these proteins are synthesized, generally they are from the ribosomes, they are usually in the form of the primary structure of the protein, that the linear change of the amino acid. So those proteins, they have to be actually undergo what we call post-translational modification. And part of that post-translational modification is actually, like, for example, in this case, when we have protein glycosylation. So protein glycosylation is one of the process, or is one of the steps, or is one of the types of post-translational modification. So it means that in this case, glucose 6-phosphate can be added into a primary proteins. So it means that the glucose 6-phosphate can be used in post-translational modification. And at the same time, these glucose 6-phosphates can be partially degraded. So these glucose 6-phosphates at the same time, glucose, these glucose 6-phosphates can undergo or can be degraded through the process of glycolysis into what we call pyruvates. It will be produced pyruvates, and these pyruvates will now be transported into the mitochondria. And in the mitochondria, using pyruvate dehydrogenase complex, then it will now break it into what we call acetyl. It will break it into acetyl-CoA. And this acetyl-CoA can be used either in TCA cycle. The acetyl-CoA generated, this acetyl-CoA generated can be used in TCA, that is tricarboxylic acid cycle, or we call it citric acid cycle. And at the same time, it can undergo lipolysis. Lipolysis, that is fatty acid metabolism. So it means that, ladies and gentlemen, the glucose 6-phosphate that is generated during glycolysis or at the first stage of glycolysis or from the glycogen, it can be used in post-translation modification. So it means that the post-translation modification is one of the fates of this metabolic pathway. So it means that when you have glucose 6-phosphates, it depends on the body demand. It depends on what body needed at that specific time. So if the glucose 6-phosphate is needed for protein glycosylation, then glucose 6-phosphate will go there. And if there is no proteins synthesized at that time, so the glucose 6-phosphate can be utilized, because if there is no energy, if there is low energy in the body, then what will happen? You need to get energy. And that is why the glucose 6-phosphate will now be partially degraded into the pyruvate here. And the pyruvate will transport into the mitochondria, and then using enzymes called pyruvate dehydrogenase complex, using pyruvates, dehydrogenase complex, pyruvate dehydrogenase complex will now break the pyruvate into acetyl-CoA, and then acetyl-CoA can undergo TCA cycle to generate ATP, or fatty acid oxidation to generate ATP. So that is one of the pathway that glucose 6-phosphate is utilized. So it depends, if there is no energy in the body, then the glucose 6-phosphate will go there. And at the same time, there is synthesis of other sugars. So there are some of the sugars like cellulose, like peptidoglycan in some microorganisms like bacteria or fungi. This glucose phosphate can be used at the same time. This glucose phosphate can be used at the same time to synthesize complex, to synthesize complex carbohydrates. And this complex carbohydrate, it can be cellulose, which can be used to synthesize the cell wall of the bacteria, or it can be peptidoglycan. So these are actually some of the importance of this glucose phosphate. And at the same time, like E. coli can use it as a carbon skeleton. It can use it as a carbon skeleton. So, ladies and gentlemen, this is just to tell you a simple example of how glucose phosphate can be utilized in different pathways, but it depends on the body demand. It depends on what body needed at that time. So we should understand. So that is the essence of metabolic regulation. Okay, there is a need for post-translation modification. Then the glucose will be released or will be actually synthesized or will be generated. And then it will now send for the post-translation modification. And if there is no post-translation modification of a particular protein and you are in need of energy, then the glucose phosphate will channel, continue with the glycolysis, generate pyruvate, and then the pyruvate will now be partially degraded using enzymes called pyruvate dehydrogenase complex, and then acetyl CoA will be generated, and the acetyl CoA will undergo fatty acids metabolism and then electron transport change. And then from there, you will have a complete, from there, you will have a complete energy generated. And of course, if the body is not in need of translation modification, no need of energy, then of course, the cellular glucose phosphate will be actually used in synthesizing complex carbohydrate molecules like cellulose, peptidoglycan, glycogen. Of course, in humans, we store glycogens as part of the carbohydrate storage form. So this is what actually happened, ladies and gentlemen, and that is the reason why we need to actually control metabolic process so that at least when we need a particular product, then it will be generated, and when we don't need that product, then we don't need to generate it. So that is the concept of metabolic regulation. Okay, and then so next time, or the next thing is that in a typically eukaryotic cell has the capacity to make 30,000 different proteins. And these 30,000 different proteins are used in catalyzing thousands of different reactions involving 100 metabolites and most shared by more than one pathway. So we should understand that, ladies and gentlemen, in typically eukaryotic cell, it can be human or any other organisms, but depends the complexity of the organisms. And of course, in some cases, the higher the complexity of the organisms, it means that the more the complex the DNA will be. And of course, the more the complex the DNA is, there is a likelihood there are so many number of genes that are found. And those genes that are found are actually responsible for synthesizing a particular protein. So that is why here, if we could understand that, yeah, in a typical eukaryotic organisms, at least that cells will have the capacity to produce like 30,000 different proteins. And each of these 30 proteins is responsible to catalyze the thousands of different reactions, which involve the use of hundreds of metabolites. So that is why we need to understand that just in just very short period of time, a high amount of a particular protein will be generated, and it can be enzymes. And when this happened, those proteins will be utilized in catalyzing catalyzing thousands of reactions. So as a result of that, those thousands of reactions or hundreds of reactions that are catalyzed by these proteins, so they have to also be properly regulated. And that is why if you look at this, this is a metabolic math. Look at this metabolic math. This metabolic math, if you can see it here, all these things that you are seeing are different reactions. Different reactions like carbohydrate metabolisms is here, protein metabolisms is here, amino acid metabolisms is here, metabolisms of cofactors and proteins are there, biosynthesis of secondary metabolite, amino acids, metabolisms of nucleotides, metabolisms of complex lipids, biodegradation of xenobiotics, metabolisms of complex carbohydrates. So all these are there. So there are thousands of reactions because if you state carbohydrate metabolisms, even within carbohydrate metabolism, there are so many pathways. So there are thousands of reactions. There are thousands of reactions that are catalyzing different types of reactions in the living system. And at the same time, these enzymes or these proteins that are generated are important in regulating the metabolic pathway. Okay. So now, difference between regulation at whole cell level and metabolic level. Yeah, because now we are talking about the metabolic regulation at whole cell level. So what is the difference between metabolism at whole cell level and at metabolic level? So a whole cell regulation affects the net change in the metabolic flux due to external changes in contact by a cell. So if you have a particular cell and there is any external changes, maybe the environment where the cells are found, there is a problem, or there is something, it may not be a problem, maybe something new is introduced into the environment and the cells will interact with it. Then as a result of that, there will be a changes. So a whole cell regulation is a type of regulation that depicts the net change in the metabolic flux. When we said metabolic flux, like for example, let's say this is the hepatocyte. And in that hepatocyte, you now have, or the cells, which is the hepatocyte, is now exposed to a toxic environment. And of course, the whole in the case of whole cell regulation, the cell will sense that external factor. And as a result of the sensing that of external factors, then there will be an increase or there will be a change in metabolic flux. And it can be to channel the whole cells of the liver to start generating antioxidants, anti-toxic proteins or anti-toxic enzymes. Like for example, in the case of xenobiotics, xenobiotics are foreign substances that get into the body and you don't need them. Because when you have something foreign into you, especially drugs, they have side effects. So when you take drugs, it means that the metabolisms or the enzyme system of detoxification of those foreign substances, those proteins that are involved in doing that, their action, their activity, as well as the amount of that enzymes may increase. So if you have increase in that, it means that there will be an increase in the metabolic flux of xenobiotic detoxification or through the detoxification system. So an external substances that introduce into the environment, it can cause a problem. And as a result of that, the cell will sense that. So once the cell senses that, then there will be a change in the metabolic flux. So that at least at least the cells will now develop a strategy that will make the cell itself to protect itself. So this is just an example. So there are many circumstances where you will have this kind of cell or whole cell regulation. So we are talking about the regulation of all the activity of a particular cell. So it is the net result of individual metabolic reactions. So if you see a cell is functioning well, it means that that function is as a result of the proper actions of the individual metabolic reactions. And that is where there is a good control, or there will be a standard control of a whole cell regulation. Because if the individual metabolic reactions are normal, it means that the whole cell should also be normal. And if there is any change that causes a problem of a particular metabolic reaction, then there's also a problem, because this whole cells will now sense that I need to now try to develop a strategy that will make it to control the situation. So that is when we are talking about the whole cell regulation. So and then the next thing is regulation at metabolic level. So specifically account individual reactions that lead to overall metabolisms or condition of a cell. So assuming now we are just looking, let's take, for example, glycolysis. In glycolysis, there are 10 steps. There are 10 steps. So you can control the whole glycolysis using an individual reaction. Of course, we have control points in the case of glycolysis, and these control points include step 1, step 3, and step 2. That is where we have that steps in glycolysis, we call them irreversible steps. And they are the control points. Because the first one is hexokinase, and the third one is phosphofructokinase 1. And the last one is the pyruvate kinase. So these are the 3 points in glycolysis that are called irreversible steps. And they are the committed steps, they are the control center. So it means that if you actually change the activity of these 3 enzymes, because they are the control points, it means that all the activities of that particular cell, all the activities of that particular glycolytic pathway will be normal. So with these 3 points, you can increase the rate of glycolysis in a cell, and you can also decrease the rate of glycolysis in some cell. So what is actually the result of these individual metabolic reactions that are found in different cells? In different maybe organelles of the cell. So when you add all those individual reactions that are taking place in different part of the cells, so when you add all this, it's what will form the cell. So that's when you will have a whole cell regulation. So we should understand that a whole cell, we are talking about the whole cell, which actually came as a result of the individual reactions in that particular cell. And then when we are talking about regulation at metabolic level, it's just to control like 1 or 2 enzymes in one particular pathway. So that is the difference between whole cell regulation and metabolic level regulation. So now we are going to look at the regulation of glycogen. What is glycogen granules? When we say glycogen granules are complex, which actually aggregates the glycogen and the enzymes that synthesize and degrade it. So the enzymes, or the complex that made of the enzymes and the glycogen itself, is what we call glycogen granules. And the general mechanisms for storing and mobilizing glycogen are same in liver and muscles. So generally we have 2 organelles in our body where glycogen are stored and mobilized. So these organelles, we have liver and muscles. So glycogen can be stored in liver and muscles. But the enzymes differ with different roles in both tissues. But the enzymes that are found in the liver and that of the muscles are same. In gut, glycogen is converted to free glucose. And this is happening with a different set of enzymes. And at the same time, glucose 1 phosphates, which is the end product of the glycogen phosphorylase reaction, is converted into glucose 6 phosphate by phosphoglucosamines. Yeah, actually in glycogen metabolism, if you have a situation where initially you eat and after you eat you have enough energy, and that enough energy you have allow the system to convert the excess glucose into glycogen using an enzyme called glycogen synthase complex. So when you have excess glucose, when you eat and you produce enough energy and you have some other remnant of glucose which are not actually utilized either in protein like glycation or any other things. So that glucose will now be channeled, transported to to the liver or muscles. So in a situation where you have when you have low concentration of glucose in your body, then what will happen? The glycogen, the glycogen, use the glycogen you stored in that liver is the one that would be that would be breaking down within what you call within an enzyme called glycogen phosphorylase. Within an enzyme called glycogen phosphorylase. That is the name of the enzymes. And what these enzymes will do to start breaking down, chopping off these complex glucose, glycogen molecules to be breaking, cutting glucose 1 phosphate. Glucose 1 phosphates. And then an enzyme called phosphoglucosamines, because our cells usually cannot recognize these glucose 1 phosphates. So what will happen? An enzyme called phosphoglucosamines now transmutates or change the position of phosphates in the glucose 1 phosphate from position 1 to 6. So after the action of this phosphoglucosamines, we will have glucose 6 phosphates. And of course, from the previous slides, I told you how this glucose 6 phosphate will be utilized in different pathways, as well as it is important in some other life processes. Okay? So now, in a skeletal muscles, it serves as an energy source to support muscles contraction. So, glycogen serves as energy sources to support muscles contraction. Because in muscles contraction, you need enough or you need a lot of energy. And of course, the glucose or the glycogen in the muscles, the glycogen in the muscles is usually not utilized. It's usually not utilized to contribute blood glucose because the lack an enzymes called glucose, like an enzyme called glucose 6 phosphatase. Why? The reason is because the glucose you use in the blood is free glucose, not glucose 6 phosphate. So the muscles doesn't have this enzyme glucose 6 phosphate. So that is why, as a result of that, due to absence of these enzymes glucose 6 phosphate, so the glycogen in the muscles is not used in maintaining the blood glucose level. And then the next thing is the glucose 6 phosphates in the liver. So it releases glucose into the blood when the blood glucose level drops. And this is actually due to the presence of enzymes called glucose 6 phosphates in the liver. So because we have this enzyme in the liver, and that is why this is happening. So these enzymes, glucose 6 phosphate is only present in the liver. This glucose 6 phosphates have glucose 1 phosphates. So after we have this glucose 1 phosphate first, have glycogen in the liver. The enzymes called glycogen phosphorylase will now release glucose 1 phosphate. Then after glucose 1 phosphate is generated, that enzymes called phosphoglucomutase will now change position of that glucose 1 phosphate to glucose 6 phosphate. How does it do that? To now add 1 glucose at carbon 6. We have glucose 1 6 phosphates. Then the phosphates group at carbon 1 will be completely removed. Then last we have glucose 6 phosphates. And then after we have the 6 phosphates, then an enzyme called glucose 6 phosphatase now remove the phosphate. And at the end, you would have 3 glucose, should be used in maintaining the blood glucose levels when it drops. Okay. So what are the metabolic pathways? So metabolic pathways, we have catabolic. Example of catabolic, we have glycogenolysis, we have glycolysis. So glycolysis and glycogenolysis are all important in baking. They break down larger molecules into smaller. Glycogen break down into glucose. Glucose is called glycogenolysis. So you break down complex glycogen into individual glucose. Process is called glycogenolysis, and the key enzymes is glycogenolysis. And at the same time, we have glycolysis, where glucose will break down pyruvate. And then in the case of anabolic process, is when you have so in the case of anabolisms, we have gluconeogenesis and glycogenesis. So in gluconeogenesis, glucose synthesized from pyruvates, and then the case of glycogenesis, glucose is used to synthesize glycogen. So this is anabolism. So this is pathways. Pathways are divided into, although if you check some other textbooks apart from catabolism and anabolism, there is also amphibolism. Amphibolism comprises both catabolism and anabolism. So when anabolism and catabolisms are taking place simultaneously, you have catabolism. So you have amphibolism. So amphibolism is when anabolism and catabolisms are taking place simultaneously. So now we are going to look at the mechanisms of regulation of key enzymes. So generally, as I said, when you have DNA, Then you have DNA molecule. So a DNA molecules is usually transcribed to produce RNA, and the RNA can undergo what they call a turnover, that is can be degraded to produce individual nucleotides. But so also at the same time, the mRNA can also undergo translation to produce enzymes. So most of the enzymes are protein, and these enzymes, generally, the enzymes, if you want to increase or decrease the rates of a particular pathway, if you want to increase or decrease the speeds, or for example, now, let's say you have glycolysis. So in glycolysis, let's say you want to, in glycolysis, let's say you want to have glucose, you have glucose, and you want this glucose to be utilized to produce pyruvate. So assuming now you need high concentration of pyruvate in a cell. So there are so many enzymes. There are so many enzymes. So how do you increase the rate of production of pyruvates? So to increase the rate of the production of pyruvates, you can actually look at it from the point of the enzymes. One, you can use what you call allosteric molecules or allosteric modulators or activators. So many enzymes, or most of the enzymes are allosteric enzymes. It means that they have other sites. They have another site apart from the active site of the enzymes. So most of these modulators or these activators or inhibitors are usually attached or are usually binded to the allosteric side of the enzymes. So once they are added, they will activate or they will increase the activity of the enzymes. So through covalent modification and allosteric regulation are the two processes that are used in increasing the activity of the enzymes. And of course, increasing the activity of the enzymes can lead to increase in turnover number, an increase who or can lead to increase in enzymes activity. So an enzyme's activity is the rate of the conversion of the substrate to product per unit time. So the more the concentration or the more the increase in activity of the enzymes, it means the faster the reaction. So through allosteric regulation, that is by using activators or inhibitors, the inhibitors or activators can bind to the allosteric side of the enzymes, which can actually lead to increase or decrease in the enzyme's activity. And there is also covalent modification. So in the case of covalent modification, it's just that we'll be adding some molecules which can be used to activate or inactivate the enzymes. And the molecules can be phosphates, can be glucose, yeah, can be phosphates, can be any other group. So it can be like methyl or phosphate. It can be added to a particular enzymes to either increase, decrease, activate, or deactivate a particular enzymes. And then there is also association with regulatory proteins. So that you can increase the activity of an enzymes by associating the enzymes with regulatory protein. You can also increase or decrease the activity of an enzyme within sequestration. So ladies and gentlemen, these are 4 ways that are used in controlling the activity of an enzyme, that is of increasing or decreasing the activity of an enzyme. So at the same time, all this post process, they are just used in increasing the activity of the enzymes. And of course, in terms of controlling the activity of the enzymes, or in the case of metabolic regulation, is not just only increasing or decreasing the activity of the enzymes. You can also increase the the rate of metabolic pathway by increasing the concentration of the enzymes. So if you increase the amount of the enzymes, the pathway, like for example in glycolysis, if you increase the concentration of hexokinase or phosphoglucokinase 1, it means that definitely the rate of metabolisms will increase. So these are the mechanisms for regulation of key enzymes. So it can be by number one, can actually regulate the enzymes by one by increasing increasing the concentration of the enzymes. And how do you increase the concentration of the enzymes? The concentration of the enzymes can increase due to increased engine expression. That is by increasing the production of messenger RNA from the DNA. So of course, if you have increase in messenger RNA, then you will have increase in the enzymes concentration. So increasing the enzymes concentration can also increase the rate of metabolic pathway. And then the second one is increase is increase is increase in enzyme is increase in enzymes activity. Is increase in enzymes activity. And how do you increase enzymes activity? Is through this four process: association with regulatory protein, allosteric regulation, covalent modification, and sequestration. So these are the four ways that you can use to increase the activity on enzymes. Okay? And all these things depend on the body demand. It depends on what are needed. It depends on what your body needed. So now, the next thing is the living cell. The living cells maintain a dynamic steady state. So when you set a dynamic steady state, generally a steady state it means it's a maintenance of constant concentration of molecules or ions in a cell or any organs of the living organisms. So when you are maintaining the concentration of a particular molecule at constant states, that is what we describe as steady states or dynamic steady states. It means that the rate of the inputs of the molecules is always equals to the rate of output of the molecule. So therefore, like for example, this is a reaction where you have A converted to S and then S converted to P. So when V1 is equal to V2, it means that the concentration of the substrate is the same, so it's constant. So it means that when we are talking about a steady state, it's like an equilibrium. But in the case of the steady state dynamics, it's just that we are trying to maintain the concentration of the molecule. Trying to maintain the concentration of the molecules. So that is why the rates in which the rate of the metabolic flux through the pathway may be high, but the concentration of the substrates will remain constant. That is what happened in the metabolic flux. You see here you have A converted to S and S converted to P. Although sometimes when steady state is disturbed by some external changes, it may be possible that the steady state can actually be disturbed by some external changes, but temporarily flux will be altered through individual metabolic pathway, so it may be altered. And this trigger regulatory mechanisms intrinsic to each pathway. But still, the net effect of all this adjustment is to turn the organisms to a new steady state. Of course, if there is a change in the environment or if there is any external factors that disturb the reaction, It may actually lead to maybe increase in the reaction, but the concentration of the substrate and product must be maintained within the required amount. Because if it exceeds the normal or below normal, there will be a problem. And that is why the concentration of the substrate must be maintained. Because homeostasis is also taking place in the cell. That is the maintenance of the internal environment. So therefore, when there is any change, of course, there may be an adjustment. But that adjustment will actually lead to the new steady state. For example, now if your body is in need of demand, or if it's in demand of a lot of glucose, of course there is going to be gluconeogenesis, so that we produce so many glucose. So, but the rates, the speed of producing the gluconeogenesis will increase. Of course, because the enzymes can be activated, increase the rate of the synthesis of the glucose through the process of gluconeogenesis. But the concentration of the glucose in the blood will not exceed the required standard value. That is what the steady state dynamic is. If there is a problem, then the body will sense it. That okay, there is the need of this particular biomolecules. So if that happened, what cells does to now increase the rates, the synthesis of the biomolecules that is needed. But when that biomolecules are being synthesized or produced, it will not exceed the standard concentration, the standard range. So that is why that is the meaning of dynamic steady state. Of course, the rate of reactions can increase, but the concentration must be maintained. And then in addition, that is actually for the flux and the steady states. So now the next thing is regulatory mechanisms evolve under strong selective pressures. Regulatory mechanisms evolved under strong selective pressures. So now, of course, sometimes some of these regulatory mechanisms usually arise when there is a selective pressure. Like for example, so what happened is like in the case of microorganisms, when you produce antibiotics, the antibiotics is a compound that kills microorganisms. So if organisms are able to sense those kinds of chemicals that kill them, the chemicals serve as a selective agents. So they are always trying to develop this strategy that they will use to protect themselves. So to control the chemicals that kills them using their own mechanisms is actually a regulatory system. So now, for example, let's say we have 4,000 genes. So that 4,000 genes produce or leads to generation of regulatory proteins. And the regulatory proteins can be receptors, can be regulators of gene expression, and maybe 500 different protein kinases. So regulatory mechanisms act over different time scales and have different sensitivities to external changes. So the mechanisms overlap. One enzyme is subject to regulation by different mechanisms. So that is why we should understand even the mechanisms that we are talking about is not applied to. So it's not the same mechanisms that you will use in controlling one particular factor or one particular mechanism. So one enzyme is subject to regulation by different mechanisms. So we should understand that in the regulatory mechanisms, there must be a pressure. Because this pressure, if there is a pressure A, the pressure A can just be maybe specific to one particular pathway and it may not affect the other pathway. And of course, the regulatory proteins that will be needed to control the situations might not be the same with the other conditions when it happens in the cell. So now, ATP using enzymes have KL between 0.1 to 1 millimolar. So ATP concentration is in typical cell, it's usually 5 millimolar. And if ATP concentration drops significantly, so it means that the rate of 100 of reaction that involve ATP will decrease and then cell will survive. So it means that the concentration, if the typical concentration of ATP in a body system, it's 5 millimolar, so it has to maintain within that. And if the concentration is dropped below 5, then it means that all that rate of hundreds of reactions that involve ATP, it would be actually decreased because there are thousands of reactions that need ATP for them to continue their processes. So now the ATP is dropped. So all those hundred of reactions will also have problem. And of course, at the end, the cell will also may die actually. Like for example, brain has no stored form of energy. So brain doesn't have any stored form of energy. So that is why glucose drops from 4 to 5 millimolar half causes mental confusion. So that is why it's very, very important because glucose is the main source of energy in the brain. So if it's below, if it goes actually below 45, it can cause problem. So five-fold decrease of glucose in the blood can lead to coma and deaths. That is just to understand that yes, a lot of things can be vitally related depending on the concentration. So if concentration of something is high, there will be a problem. And if it is low, there is also a problem. So you need to understand what effect, what metabolite is needed this time and which one is not needed at this time. And if there is a decrease of one particular metabolite, what will happen? What is the problem? So all this is important. It's important in metabolic regulation. So now we are going to look at the regulation of glycolysis and gluconeogenesis. So gluconeogenesis is performed primarily in liver. So we should understand that yes, glycolysis glycolysis is taking gluconeogenesis is usually or is primarily happened in the liver and glucose for exports for that tissues. Because when we say gluconeogenesis, it's a process where non-carbohydrate materials are utilized to generate uh, uh, glucose. So gluconeogenesis, you use something like alanine, amino acid pyruvate. You use it when you use these molecules to generate ATP. So to generate glucose is called gluconeogenesis. And the reactions of glycolysis are hexagonic, so as to be essentially reversible. So it has a negative value. Because when we said reaction have negative delta G, it means that the reaction is feasible and is spontaneous. Spontaneous. And the key enzymes in glycolysis is hexokinase, phosphorokinase 1, and pyruvate kinase. So these are the key regulatory enzymes. These are the three regulatory enzymes that are found in glycolysis, and these are the enzymes: hexokinase, phosphorokinase 1, and pyruvate kinase. And they have large negative delta H. Right. And that is to tell us that the action is feasible. Ladies and gentlemen, I'm going to stop here and then we'll continue later. Please, if you watch this video, please subscribe.